Okay, so what we're going to do today is finish off the cranial nerves, and then we're going to go ahead and get started into the brainstem section. So we'll just keep pushing ahead. So what I did last night is I did post my copy. What I did was I took all the cranial nerves and the reflexes of the cranial nerves and tried to do like a concept map of how information flows from a cranial nerve nucleus out to the periphery, from the periphery back to cranial nerve nuclei. So I put those up there just for you to have a reference, but obviously these are much more useful to you if you create your own, right? And so that helps you learn the material and think about the flow of information and what types of fibers are associated with those. So I would really encourage you to try to use those just as a reference rather than just trying to study from my concept maps, because remember we talked about that concept mapping is a good way to kind of test yourself as well to see if you know the material. And then the other thing that I posted as we go into Brainstem, so a lot of this is pretty dry because it's a lot of just plain neuroanatomy and understanding where nuclei are, where other structures are within the brainstem, so that when we have lesions to particular areas of the brainstem, we can start to put together what structures have been damaged and therefore what sets of signs and symptoms come together in that same injury at that same level. So we're gonna do the brainstem death march starting later today and continue on into next time, where we go through sections of the brainstem and start identifying structures. So as part of your exam, the next exam, you will have to be able to identify structures within brainstem sections. So there are some templates that I put up, some blank templates of that I'll be using in class, but that way you have these blank templates to use to practice labeling structures and identifying structures. So those should both be available to you in the handout section of your Blackboard. Okay, so we left off with the vestibular cochlear nerve, cranial nerve 8. Remember, that's a purely sensory nerve. Now we're going to move on to cranial nerve 9, which is the glossopharyngeal nerve. This is a mixed nerve as well, meaning it's going to have both motor and sensory fibers. But again, it's a little more complicated because it has different types of motor and different types of sensory fibers. As we go through it, we'll take a look at that. So in terms of motor innervation, we see that there are those special visceral efferent fibers, meaning they are going to innervate some skeletal muscle that derive from branchial arches. In this case, there's actually only one muscle that is innervated by these types of fibers running with the glossopharyngeal nerve, and that is the stylopharyngeus muscle. And that is a muscle that helps to elevate the pharynx. The cell bodies for those lower motor neurons that go out and innervate that muscle, they are in a nucleus that exists within the medulla of the brainstem, and that's this pink nucleus here. That is the nucleus ambiguous. Now, most of the cells in that nucleus, their fibers are actually going to run with cranial nerve 10 to innervate more of the special visceral efferents, but the fibers that are running with the glossopharyngeal nerve, they have their cell bodies in that nucleus as well. The other type of motor fibers that we see running with the glossopharyngeal nerve are going to be parasympathetic fibers. So remember, those are always general visceral efferent fibers. And again, it's a 2-neuron pathway to get out to the visceral tissue out in the periphery. So the preganglionic cell bodies will be within the brainstem. In this case, they exist in a nucleus known as the inferior salivatory nucleus. Remember, we saw a salivatory nucleus associated with cranial nerve 7. This is a different salivatory nucleus, also located in the pons though. And then those fibers leave with cranial nerve 9. Remember, cranial nerve 9 exits the skull through the jugular foramen. So outside the skull, there will be a ganglion associated with that nerve. In the case of the general visceral efferents for cranial nerve 9, the visceral tissue innervated by cranial nerve 9 is going to be the parotid gland. And so what we're going to see is again, the preganglionic cell bodies will be here in the inferior salivatory nucleus. Those fibers will leave the glossopharyngeal nerve as the lesser petrosal nerve, which runs through that infratemporal fossa. And within that infratemporal fossa, we have the otic ganglion. So that's where our cell bodies are for our postganglionic neurons. And their axons actually use a branch of cranial nerve 5 to get to the parotid gland. They use the auriculotemporal nerve to be able to get to that parotid gland. So that's a pathway out for those general visceral efferents. And again, the visceral tissue that we see innervated by those fibers is the parotid gland, which is a salivary gland. Also running with the cranial nerve 9, so those are two motor components of that nerve. There's also going to be sensory fibers running. So they're going to be bringing information using cranial nerve 9 back into the brainstem. And some of those fibers that we see are fibers that are carrying general somatic afferent, again, afferent meaning sensory information. And by general somatic, that means they're going to be carrying touch or pain or pressure. And remember, where we see some skin out in the periphery in the head area that has innervation other than the trigeminal nerve, there was a little patch outside the ear. So remember, in addition to trigeminal covering most of the face, this little patch of skin actually gets its innervation for somatosensory using cranial nerve 7 and 9 and 10. Okay, so this is where those sensory fibers would be relaying information from, at least from the skin. They also can provide some sensation from the posterior 1/3 of the tongue and the pharynx as well. So when we say sensation from the posterior 1/3 of the tongue and the pharynx, that means that you're able to register things like touch or pain on the posterior 1/3 of the tongue or on the pharynx, okay? That's how you get that afferent information back in. So that's what these blue fibers are representing. The cell bodies for those sensory neurons are going to be, again, outside the central nervous system. So they're going to be in what's known as the superior, so that's this one here, the superior glossopharyngeal ganglion. And then their central axons come in. And again, these fibers outside of the central nervous system are running with cranial nerve 9 into the brainstem. But once they get into the brainstem, they actually hand that information off to the trigeminal nuclei that are relaying information about touch and pain and temperature from the face. So that's where they hand it off, is to the trigeminal nuclei. Okay, so those are our general somatic afferent fibers. Those are some of the sensory fibers that run with cranial nerve 9. Now, the other types of cranial fibers that we see, or sensory fibers running with cranial nerve 9, one are going to be those special visceral afferent fibers. Remember, whenever we say special visceral afferent, that's referring to taste. So the other place we've seen that so far is these types of fibers running with cranial nerve 7, and those fibers provided taste from the anterior 2/3 of the tongue. The distinction with cranial nerve 9 is we have those SVA fibers, but they're relaying taste from the posterior 1/3 of the tongue, okay? So again, because these are afferent fibers, they will run back to the central nervous system. Their cell bodies, again, will be outside of the central nervous system. So in this case, in the inferior glossopharyngeal ganglion, which sits right next to cranial nerve 9, and then into the medullary area of the brainstem. And here they synapse in a nucleus known as the solitary nucleus. But remember, the whole rostral end of that solitary nucleus is dedicated to receiving just taste fibers. So all of our taste information ends up in the rostral solitary nucleus. So that's where these will relay that information. So that's shown here, this relay back. We also will get information, sensory information that also goes to the solitary nucleus, but this information ends up in the caudal solitary nucleus, and this is information provided by general visceral afferents. So these are sensory fibers that are relaying autonomic information about blood pressure and chemical composition of the blood, particularly specifically from the area of the carotid body and the carotid sinus, so right where the carotid artery bifurcates. So those afferents relay that information back again to the solitary nucleus. Their cell bodies are also in the inferior glossopharyngeal ganglion, But in this case, they will end up in the caudal end of that nucleus that's dedicated to those general visceral afferent fibers. Okay, so again, glossopharyngeal nerve has both a mix of motor and sensory components to it. And within each of those groups, we have subdivisions of those. So thinking about what types of fibers are with it. Sorry, did somebody have?
I had a question.
Yeah.
About the general afferent or sensory afferents for the ear canal or the general somatic afferent.
Yeah.
What's the differentiation between the patch of skin that it shares? Because I know you said that there's two other cranial nerves.
Yeah, there's no difference. It's just that during development, that particular area of skin outside of the ear there, it was initially associated with those pharyngeal arches that were related to those particular cranial nerves, 7, 9, and 10, rather than derive that piece of skin deriving from the pharyngeal arch that's related to cranial nerve 5. So that's the only distinction. And why that occurs, that we just get that little patch and then everything else is cranial nerve 5, that's unknown. That's just how it develops.
Okay, so all three just share the same kind of patch.
Yeah, so if you remember, let's see if we can go back. Oh, I don't know if I have that up on the slide, actually. Let me see if I can pull that up. To get a couple things out of that. Sorry, this takes a few minutes. Okay, so do you remember this picture? So it's really just this area, and then that area, it gets innervation from these 3 cranial nerves rather than cranial nerve 5. But all of these fibers, either cranial nerve 5 as well as these fibers from this area of skin for 7, 9, and 10, those all are going to dump that somatosensory information into those trigeminal nuclei, because they're the one, that's the pathway that takes somatosensation up to the brain, where you actually can perceive touch or pain or pressure, right? Did that answer your question?
Yeah, I was just wondering if there was something different between those 3.
No, it's just how that patch of skin, like what part of the developing central nervous system it was associated when the neurons first grow out and make their connections.
Okay, thank you.
Okay. Let's see. I'm gonna get out of this now. Okay, let's go back to this. Okay, so now we're going to move on to cranial nerve. Oh, wait, let's go back to cranial nerve 9. Hold on 1 second. Okay, now we're back. So with cranial nerve 9, again, knowing what types of fibers are present within cranial nerve 9, now we can be able to make predictions about what types of deficits we'd see in a patient with a cranial 9 injury. And a lesion of cranial nerve 9 is actually fairly rare to get just an isolated lesion of that one particular cranial nerve. It can happen sometimes with demyelination, pathologies, or if you had diabetic neuropathies of that nerve, but it's not a commonly injured nerve. So again, things that you would see with a cranial nerve injury would be loss of taste in the posterior 1/3 of the tongue and the pharynx, or the posterior 1/3 of the tongue, loss of sensation to the posterior 1/3 of the tongue and the pharynx and palate. Now, cranial nerve 9 is involved in a reflex that allows us to test both cranial nerve 9 and cranial nerve 10. So cranial nerve 9 is actually the sensory component of what's known as the gag reflex. And cranial nerve 10 is going to be the motor limb of that reflex. So what happens is, I don't know if you've ever had something go in the back of your throat and touch your palate or your uvula or something like that, and your reaction is to gag, your pharynx elevates. And so what happens is cranial nerve 9 relays that sensory touch information back to the central nervous system and hands it off to motor nuclei of cranial nerve 10. And that's what causes the contraction of those muscles. So by doing a gag reflex test, you're testing cranial nerve 9 and cranial nerve 10. So any loss of the gag reflex could potentially be attributed to cranial nerve 9. Also, you would see decreased salivation because of the effects on the parotid gland. So those are all losses that we might see in a cranial nerve 9 injury. Okay. Okay, so now we're going to move on to cranial nerve 10. And with cranial nerve 10, again, this is going to be a mixed nerve with motor and sensory fibers running in it. So the motor fibers are special visceral efferent fibers, meaning these skeletal muscles that are innervated by this nerve derived from brachial arches. So this is our major innervator to muscles of the larynx and pharynx. So with the exception of that stylopharyngeus muscle that we saw cranial nerve 9 innervate, all the rest are innervated by cranial nerve 10. So muscles of the larynx, pharynx, the upper esophagus, also the palate. So these are all muscles used for speaking and swallowing. So you need to think about that when we have lesions of this nerve that could impact those functions for the patient. So as we mentioned before, the nucleus that cranial nerve 9 used to innervate the special visceral efferents used by cranial nerve 9 derived from a nucleus here in the medulla known as the nucleus ambiguous. And we see the same for cranial nerve 10. It's that same nucleus that houses the cell bodies of those lower motor neurons that then leave with the vagus nerve. Again, they go outside the skull, they descend through the neck, and as they're descending, they innervate muscles of the larynx and pharynx. The other motor component we see is as well general visceral efferents. Again, those are parasympathetic fibers. So the preganglionic neurons are going to be located in a nucleus known as the dorsal motor nucleus of the vagus nerve. So that's an easy one to remember because it gives you the name of the nerve that those fibers are running with. So those preganglionics leave with the vagus nerve. Their destination is to descend through the neck and down into the thoracic and abdominal cavity to innervate the viscera in the thoracic cavity and most of the viscera in the abdominal cavity. The postganglionic neurons, remember with the vagus nerve, because it's parasympathetic, the preganglionics are much longer than the postganglionics. So the ganglia for the postganglionics are within the walls of the organ. That's where they synapse in what we call terminal ganglia. So that's our output. That's our motor output for vagus nerve. For sensory components of vagus nerve, similar, actually, I'm going to hop ahead to the next slide. Similar to what we saw with cranial nerve 9, there's just that little patch of skin outside the ear where we receive touch and pain and temperature and pressure. So again, these are afferent neurons, sensory neurons, so their cell bodies will be outside of the central nervous system. In this case, they are in the superior vagal ganglia, which sits alongside the vagus nerve. And then their central axons carry that information again into trigeminal nuclei. The other place that's innervated though by fibers, general somatic afferent fibers of cranial nerve 10 are going to be the tympanic membrane, but also the meninges for the posterior fossa. Okay, so we see those as well. And then the other sensory information being relayed with the cranial nerve 10, similar to what we saw with cranial nerve 9, we have those special visceral afferents. Those are taste fibers again, but this case, they're relaying the sensation of taste from taste buds that are actually located back in the pharynx and on the epiglottis, the mucosal covering of the epiglottis of the larynx. So those again are sensory neurons. So their cell bodies are going to be in that inferior vagal ganglia, so down in here, and then their central axons will relay that information to, remember, taste always goes to the rostral solitary nucleus. We also will have some general visceral afferents, so again, relaying information particularly about blood pressure, chemical composition of blood, but in this case, instead of from the carotid body, like our carotid sinus area, like cranial nerve 9, that information gets relayed from the aortic arch, okay? And again, the cell bodies are going to be in the inferior vagal ganglia. And in this case, because this is general physiological information or visceral afferent information, that's going to go to the caudal solitary nucleus, okay? So those are our fibers that we see present in cranial nerve 10. So when we would have lesions of the vagus nerve, we think about, again, the types of fibers contained in the vagus nerve. If we lesion that visceral branch that passes down through the neck and is destined to innervate viscera in the thorax and abdomen, then you could have things like transient tachycardia, right? Because the parasympathetic effects on the heart via the vagus nerve are to slow heart rate down. So if you damage those, then you have the sympathetics unopposed. So you'd see increases in heart rate. You may see difficulties with breathing because of effects on innervation of visceral structures associated with the lung. But particularly what you would see is if you had damage to that skeletal branch, so the fibers that are going out to innervate muscles of the larynx and pharynx. One way we could see if there is an effect is looking at the area of the soft palate. So the soft palate's muscles work in such a way that they kind of arch over towards this structure here, which is the uvula, which hangs right down the middle of the soft palate here at the back of the throat. When the pharynx's soft palate is working correctly because those muscles innervation coming from cranial nerve 10 is intact, what happens is those two arches lift together on either side. And so you get a raising of the uvula right up the midline of the back of the throat there. It goes straight up. What happened in this picture, if you ask the person to, and that happens, you can see that if the palate is working correctly by just having the person open their mouth wide, shining a light back there and asking them to say ah, because ah creates that lift in the palate. What we're seeing in this picture is on the person's left side, first of all, you can see that the soft palate is hanging down lower than the right side when they're asking the patient to say ah. The right side is lifting up, but the left side is hanging down lower because this left side doesn't lift to the same degree as the right. The right side actually pulls that uvula towards the right side. So that uvula ends up deviating to the unaffected side, and that tells you that there's a weakness on the side that's away from the uvula. So that's one way we can test cranial nerve 9. The other is to just do the gag reflex itself, where you go in and touch the palate to see if you get, again, that bilateral response of the soft palate raising. One thing that we do know, because the bulk of the muscles of the larynx and pharynx are innervated by cranial nerve 10, is a lesion to that causes what we call the 3 Ds. So the person may experience dysphonia because of branches that go to the larynx and innervate musculature of the larynx. So they may have a hoarseness of their voice. They could have dysphagia, so they could have difficulties with swallowing because of innervation of the palate and the larynx and the pharynx. And then they could also have dysarthria, which is a difficulty with articulating words. So their words kind of sound slurred. So these things all can be attributed to weaknesses of those muscles of the laryngeal and the pharyngeal. Now, some of these things happen with other lesions, but we just are aware that these are effects that could happen with a cranial nerve 10 lesion as well. Again, isolated lesions of cranial nerve 10, just like cranial nerve 9, are relatively rare, but again, similar things could cause those. Demyelination or diabetic neuropathy can contribute to those. So these are some videos from the neurologic exam. Again, these don't have sound associated with them. I think the first one, he is asking him to say ah, but let me see. Yeah, ah. Okay, so is that palate lifting up equally on both sides in that video? Okay, which side is it raising to?
The left.
The left side, right? And the uvula was deviating over to that side too. So that's showing some weakness here in the right side. And I think the next video, they're doing a gag reflex. So they're touching to see what happens back there. It's a little bit harder to see. Yeah, so I think they're touching over the left. So you can see how that the left side is raising up really high, and the uvula is kind of swinging over here a little bit to the left. Okay, we're getting there. 2 more nerves down, or 2 more to go. So the next nerve is cranial nerve 11, which is the spinal accessory nerve. So this nerve is actually considered in your book to be special visceral efferent fibers that innervate skeletal muscle. So this is a purely motor nerve. They innervate musculature of the sternocleidomastoid and the trapezius muscle. Those are the 2 muscles that they innervate. Now, what's interesting about this nerve is that it actually derives from the spinal cord. So these are motor neurons that are in the spinal cord, but in this case, they're not coming from the ventral horns of the spinal cord, and they come from the cervical area of the spinal cord. So from C1 to C6. They actually, their cell bodies are here kind of right in this intermediate area, or at the base of the dorsal horn, getting into the intermediate areas where the cell bodies are. And they don't leave with the ventral root of the spinal cord. They actually come directly off laterally. So they emerge laterally out of the spinal cord in that cervical area. They start to come together. So as C6 and C5 and C4 fibers come out, they come together and they form a structure that runs up into the foramen magnum. Okay, so they're coming from the cervical area of the spinal cord. They go up into the skull through the foramen magnum, and they're in the posterior fossa of the near the brainstem. And then what we see is they actually hop a ride on the vagus nerve after it emerges from the brainstem. And they just run with that vagus nerve just a little bit. And then as that vagus nerve exits, or as the cranial nerves exit the jugular foramen, then what we see is this nerve comes off as a separate nerve out the jugular foramen with cranial nerve 9 and 10. So it's called a cranial nerve because it actually emerges from the cranium, but in reality, its origins is not from the brainstem. Its origins are here from the cervical spinal cord. It goes up into the cranium and uses a cranial nerve to get over to the opening where it will then emerge from the skull, okay? But in any case, those are considered special visceral efferent fibers, and they innervate the trapezius and sternocleidomastoids. So you have to think about weakness of the actions of those muscles in terms of lesions. When we see lesions of that nerve ipsilaterally, it would result in an ipsilateral paralysis of those muscles. And the most common way that we see that nerve damaged is actually in surgeries to remove lymph nodes. Particularly, there's a chain of lymph nodes here along the sternocleidomastoid, or any surgery into that posterior triangle area of the neck could cause damage to that spinal accessory nerve. So this is just going to show the weakness. And again, because these are lower motor neurons, what you can see over time with damage to this nerve is atrophy of those muscles. So this is the same guy again. Obviously, his right side is more impaired than his left. So they're going to have him kind of contract his neck against resistance. And you can see especially the sternocleidomastoid more pronounced here than we see over on the right side. So you can see that tendinous insertion. And this is from behind, showing a normal bulk for the trapezius muscle that comes over the shoulder, but you see quite a bit of atrophy over here on the right side. Okay, so he's had a lesion of his right spinal accessory nerve. Okay, and now the last nerve, cranial nerve 12, the hypoglossal nerve. So this is a nerve that innervates the musculature of the tongue. These are lower motor neurons. It's a purely motor nerve, but these are general somatic efferent fibers. So again, these derive from those occipital somites. These muscles, the lower motor neurons are localized in a nucleus within the medulla of the brainstem, known as the hypoglossal nucleus. Those fibers leave with cranial nerve 12, and they innervate the muscles of the tongue. When we see lesions to ipsilateral lesions to the hypoglossal nerve, one of the main muscles that gives us the ability to protrude our tongue out of the mouth is the genioglossus muscle. And it's this big fan-shaped muscle, and you have one on either side that those fibers kind of come together in the midline. And so as they contract, they actually kind of push against each other, and that helps to push the tongue straight out forward, okay? If one of these muscles is weak, what happens is, say this muscle is weak, this muscle now, it still protrudes the tongue out, but it's gonna, because it's stronger than the other muscle, it's gonna push it over to that weak side, to that affected side. So what happens when a person has a cranial nerve 12 lesion, one of the things you do is you ask them to stick their tongue straight out, and they should be able to keep that midline position of the tongue. If there's weakness on one side or the other because of lesion to the hypoglossal, what will happen is their tongue will deviate over to that affected side. So the way you can remember that is you say that the patient licks their lesion. All right, their tongue is going to go over towards the side where the lesion is. Okay, and that happens in a lower motor neuron lesion of hypoglossal. So this is just a video showing, just licked all over my face, showing a hypoglossal lesion. And one of the other things that we see, again, because this is a lower motor neuron lesion, is you may see atrophy of the tongue. There'll be weakness in the tongue on the side of the lesion. You may see atrophy of the tongue on that side. Again, when they ask them to stick their tongue out, it'll deviate towards the weak side. And sometimes you'll also see fasciculations of the tongue on that weak side as well, again, because it's a lower motor neuron injury. So we'll take a look at this patient.
Just get your tongue all the way out. Again, we're just gonna.
So you can see that this person, again, has an injury. This is a different patient, but on the right side, right? Because his tongue is protruding out into the right. We also see some significant atrophy here on this right side.
As far as you can. You see if you can get all the way down to your chin. Good. I want you to move your tongue over here to the left, to the right side. I want you to move over to the left, over here again, and over here again.
So he's much stronger. You can get it further over when he pushes with that left side.
I want you now to stick it all the way out. I want you to push against the tongue blade.
And if you watch carefully, you'll see the fasciculations here on the tongue. It's almost like a little bag of worms swarming around.
Okay. And only push it back. Push it back. Okay, and over here again. Only push it back. Push it back. Okay, good.
Okay. Okay, so that finishes up our discussion of cranial nerves. Okay, so you should be familiar with the, again, the naming, the nomenclature of the cranial nerves. You need to know the types of fibers and the nuclei associated with both sensory and motor fibers for each of the cranial nerves. And then given the collection of fibers within a cranial nerve, you should be able to recognize if a person has a group of signs and symptoms, what cranial nerve might be involved, or with a cranial nerve lesion, be able to indicate what types of deficits that person would have with that cranial nerve lesion, okay? Okay, so what we're gonna do now is move more centrally into the brainstem. And again, we'll revisit the cranial nerve nuclei that we have mentioned with each of the cranial nerves, but now we'll have a better understanding of what parts of the brainstem each of those cranial nerve nuclei are associated with. But in addition, we're going to be looking at other structures that help to localize a lesion to a particular area of the brainstem. So things like the motor tracts and also some other nuclei that may be unique to a particular area of the brainstem. So we'll do a little bit of review on the brainstem first. So let's see. Okay, so hopefully this picture by now looks pretty familiar to you. So what we're going to do first is review some of the growth structures on the brainstem, and then we'll start looking at internal structures. In terms of what is the brainstem, remember from caudal to rostral, it consists of 3 separate areas. We have the medulla down here. So first of all, is this a ventral or a dorsal view over here on the left?
Ventral.
That's a ventral view, okay? And there's certain things that tell me that, okay? So if we look at this most caudal area, the feature that tells me I'm in the ventral surface is the presence of this thick fiber band on either side of the midline. Those are my medullary pyramids, okay? So those stick up right in the center. And then we have just in the rostral area of the medulla, we have another lump that kind of sticks out here that's known as the olive. And there's a nucleus under there that creates that olive. So this underneath here is a nucleus. What's running here are some descending motor fibers, okay? So this area here is the medulla. Just rostral the medulla is this area where we see these big bulges on either side. That's our pons. And then just rostral to that, we again have two thick fiber bands. These are motor fiber tracts. Actually, they're the same fiber tract that we see down here in the pyramids. And those make up the cerebral peduncles, and they have a depression between them, which is our interpeduncular fossa. So all of this is our brainstem, medulla, pons, midbrain. And then these big structures up here are actually part of the diencephalon. Those are the thalami on either side. We mentioned that one thing that is unique about the brainstem is the presence of cranial nerves communicating information from the periphery back to the brainstem. So most of the cranial nerves can be viewed on this ventral view because they come off ventrally or ventrolaterally off the brainstem. So several of those cranial nerves are going to be associated with the medullary area here caudally. So the cranial nerve 12, that emerges from the medulla. It comes out right between the pyramid and the olive. We have the spinal accessory nerve. It's going to be lower here along the ventrolateral surface of the medulla, because remember that actually is coming up from the cervical area of the spinal cord. And then we have a little bit higher here, just lateral to the olive is going to be our vagus nerve, okay? And then most rostrally here in a most lateral position will be our glossopharyngeal. And then we can see several nerves running right at that pontomedullary junction. So from lateral to medial, we had first the glossopharyngeal, the vestibular cochlear, cranial nerve 8, the facial, cranial nerve 7, and then the abducens, cranial nerve 6, okay? Now, cranial nerve 7 and cranial nerve 6 actually have their nuclei in the pons. Cranial nerve 8 kind of spans between the pons and the medulla, and the glossopharyngeal nerve is mostly associated with nuclei in the medulla, okay? And then in terms of the pons, one cranial nerve that is prominently associated with the ventrolateral surface of the pons is the trigeminal nerve. So we see that being associated with mid-pons level. And then the midbrain, there are two cranial nerves associated with the midbrain, cranial nerve 3 and cranial nerve 4. Cranial nerve 3 actually is seen here on the ventral view. It comes right out at this interpeduncular fossa between the two cerebral peduncles. Cranial nerve 4, we see that coming around laterally. Remember, that is the only cranial nerve that exits the brainstem from the dorsal surface, and then it wraps around here laterally to make its way to a ventral position. So those are the cranial nerves that are actually directly associated with the brainstem. Remember, there were two other cranial nerves. One of them we see with this dissection here, and that is cranial nerve 2. That's the optic nerve. So that's relaying visual information from the eyes, but that doesn't have a direct connection to the brainstem. Those optic tracts will then direct that information to a nucleus of the thalami, okay? So those are structures that we can see and be aware of on the ventral view, at least of growth structures on the ventral view of the brainstem. When we look at a dorsal view of the brainstem, what has happened here is we have removed the cerebellum so that we can see the different parts of the brainstem. Remember, the cerebellum sits over the top of the fourth ventricle, which spans between the pons and the medulla. So where we've cut the cerebellum away are these areas here, which are the cerebellar peduncles. Those help to connect the cerebellum to different areas of the brainstem. So we've cut that and removed that. We see this triangular area here, which is our area of our fourth ventricle. Again, we should be able to still make out the three areas of the brainstem. So down here, most caudally, almost to the points of the triangle here, we have our medullary area. And again, structures that are going to allow us to define that medullary area. Coming from the spinal cord, we had those dorsal columns, the gracilis and the cuneatus. And so we'll see some expansions here at the caudal medulla where nuclei associated with those tracts are located. So the nucleus gracilis and the nucleus cuneatus would be just lateral to that. In terms of the pons, we don't really see a lot associated with the pons other than these cerebral peduncles. There is a bulge here on the floor of the fourth ventricle. It's called the facial colliculus. That is a structure that is associated with the pons as well. So in this area up in here, kind of from the top part of this triangle, this should be our area of the pons. And then rostral to that, we have these 4 little bulges on the dorsal surface of the brainstem. They demarcate the area of the midbrain. And so those 4 little bulges are the colliculi. We have superior colliculi, inferior colliculi, and those together make up an area known as the tectum, and that's on the dorsal surface of the midbrain. And this is where we see those trochlear nerves coming off of the dorsal surface of the brainstem. It's the only cranial nerve that comes off the dorsal surface, okay? So you should be familiar with some of the landmarks and gross features to be able to identify different areas of the brainstem from a dorsal and a ventral view. So in terms of just anatomical areas of the brainstem as a whole, so not particular areas of the brainstem, but the brainstem as a whole, there are 3 different types of anatomical areas that we see in the brainstem. So remember that the running through the brainstem is going to be parts of the ventricular system, which are continuous with the central canal, the spinal cord, caudal to the brainstem. So there's a narrow channel running through the midbrain area. That was the cerebral aqueduct. Then that opens into that big triangular shaped structure that we saw in the dorsal view, which is the 4th ventricle. And then it narrows down again as it runs through the caudal medulla and into the spinal cord. So this is our ventricular system. The bulk of the brainstem that sits ventral to the ventricular system is what we would call the tegmentum, okay, of the brainstem. So this is really where the bulk of the structures of the brainstem reside. And that includes our long tracts that are carrying motor and sensory information from the spinal cord up to higher centers and from higher centers back down to the spinal cord. That's where we're going to see most of those long tracts. Our cranial nerve nuclei will be embedded within this tegmental area. And then we'll see some other nuclear structures and some other networks, like something known as the reticular formation, are also making up this tegmentum. So it really makes up the bulk of what we have for brainstem tissue. Now, on all areas of the brainstem, on the ventral surface of all areas of the brainstem, that is basically comprised of a descending motor tract that originates up in the cerebral cortex. That's the corticospinal tract. It's a tract that starts in the cortex and is making its way down through the brainstem to eventually end up in the spinal cord. So those motor fibers running down, they exist on this ventral surface. So that's referred to as the basis of the brainstem. So that's the ventral area of each part of the brainstem, the midbrain, the pons, and the medulla, okay? That's basically made up of, well, you should say corticospinal and corticobulbar fibers, because some fibers will peel off in the brainstem to innervate lower motor neurons. So these are all upper motor neurons running in these tracts. So in the area of the midbrain, those descending basis fibers form the cerebral peduncles. In the area of the pons, those form the basilar, or help to form the basilar area of the pons, together with pontine nuclei. And then in the medullary area, they coalesce and form the medulla. So it's all one tract. It makes up the ventral surface in every area of the brainstem. And then there's a third anatomical area that we see that is actually dorsal to the ventricular system. And this is known as the tectum. And the only place we see this anatomical area is in the midbrain. It forms the roof of the midbrain. And again, that's where we find the superior and inferior colliculi. So we have here a cross section of the midbrain so that we can show each of those anatomical areas in a cross-section view. So again, what we see, here's our cerebral aqueduct, and the bulk of the brainstem that sits ventral to the cerebral aqueduct is this area here, the tegmentum. And in this tegmentum, there are going to be several structures. There are going to be long tracts that are motor and sensory tracts running up and down the brainstem. There's going to be cranial nerve nuclei, and then there's going to be other nuclei like the red nuclei associated with cerebellar function, or the substantia nigra associated with basal ganglia function. So there's other structures in here as well. This area here is the basis, and the only thing there are going to be these descending upper motor neurons that are relaying that upper motor neuron information to lower motor neurons, either in the brainstem or the spinal cord, okay? So that's kind of just an overview of anatomical demarcation within the brainstem. So this is just listing the structures that we said we see within the brainstem. Again, ascending and descending tracts will run in the brainstem. We'll have cranial nerve nuclei within the brainstem. We do have fiber tracts that communicate information between the brainstem and the cerebellum. So those are the cerebellar peduncles. Those will be associated with different areas of the brainstem. And then also running through, it's hard to point them out on a cross section, like a Silverstein section, because they're somewhat diffuse, are going to be the descending autonomic pathway. So remember, we talked about with the visceral motor output of sympathetic and parasympathetic, it's a lower motor neuron pathway out that's a 2-neuron pathway, right? A pre and post ganglionic neuron. Those make up the lower motor neuron pathway. But those lower motor neurons, those preganglionics, their activity can be modulated by descending projections from the hypothalamus through the brainstem and down into the, or onto parasympathetics in the brainstem, or onto sympathetics and parasympathetics in the spinal cord to regulate their activity. And then there's a structure that is a very diffuse collection of nerve cell bodies and axonal projections that is referred to as the reticular formation. And the reason for that, reticular means net, and that's again because it's not going to be always this very clearly staining structure. It's kind of diffuse and runs through the core of the tegmentum. So we don't point to a specific structure in the brainstem when we say reticular formation, but we kind of point into the tegmental area and say the reticular formation is running through this area. So this is a sagittal view, again, reviewing some of those things that we would find within the brainstem. And so again, because we know that these structures exist in the brainstem, we can make predictions about what types of deficits a person would have when they have a brainstem lesion. So one thing that we see is that embedded in the white matter in the tegmentum all along the brainstem, each part of the brainstem had some cranial nerves associated with it. We will see nuclei associated with those different cranial nerves, right? So that's something that's unique to the brainstem and to different areas of the brainstem. Certain cranial nerves have their nuclei in different parts of the brainstem. The other thing that we mentioned is that there are these long vertical tracts. So the tracts carrying somatosensory information about touch or pain, or the motor tracts carrying voluntary motor activity down. Those are in the brainstem as well. So we could have vertical tract signs with a lesions to the brainstem. So we could have cranial nerve signs if we lesion the nuclei associated with the cranial nerves. That's unique to the brainstem. We could have vertical tract signs if we lesion those ascending and descending motor and sensory tracts, but that doesn't necessarily localize lesion to the brainstem, right? Because we can see vertical tract signs in the spinal cord as well. How those vertical tract signs present may be able to give us some information. But for example, if it's the whole body, then we know we're above the level of the spinal cord, but that wouldn't necessarily distinguish a lesion in the cerebrum either from the brainstem. And then the other thing that we mentioned, there are cerebellar peduncles that are going to attach the cerebellum to different parts of the brainstem. So one thing that we can see with a brainstem lesion is the person could exhibit ataxia. So remember, we could see ataxia if the cerebellum itself was lesioned, but if you lesion those input and output pathways to the cerebellum, that's the same as lesioning the cerebellum. So you could have ataxia associated with brainstem lesions as well. And then finally, there is this loose network of neurons, cell bodies, and axonal projections, which is together referred to as the reticular formation. And the reticular formation has a lot of different functions because it will have kind of separate little nuclei. They're just more diffuse nuclei. They're not as compacted as we've seen before. One very important function of the reticular formation is it sets up our level of alertness, that we are awake, basically, okay? And that we can pay attention to sensory information coming in to the environment. So our level of consciousness is very dependent on an intact and functioning reticular formation. We've talked about, when we talked about somatosensation, that the reticular formation can also play a role in pain modulation. So remember, there were projections from the periaqueductal gray or nuclei in the reticular formation that will feed back down onto the dorsal horn of the spinal cord and help modulate the relay of pain information. We've also talked about how the reticular formation could have effects in regulating tone of musculature, particularly proximal musculature, because we saw some reticulospinal tracts when we covered the motor systems, okay? And then we're going to talk a little bit about, when we get into the visual system as well, that the reticular formation can also play a role in coordination of eye movements and vision and control of gaze centers. And then also embedded within the reticular formation are going to be groups of neurons that are very important that form central pattern generators, for example, for breathing, so rhythmic breathing, or for cardiovascular function. So there's some autonomic activity that gets controlled by the reticular formation as well. So even though it's not a very distinct anatomical entity that we can really pinpoint and point out specifically within the brainstem, it has very important roles in terms of function of the brainstem. So just to simplify it a little bit, because we're not going to go through all the individual parts of the reticular formation, functionally, we can kind of classify the reticular formation into a rostral area, the reticular formation, and a caudal area. So the rostral area is indicated in blue, shaded in purple, and the caudal area is shaded in green. And again, remember, this just kind of exists within this tegmental area, diffuse within the tegmental area. The rostral area usually extends from about the mid pons all the way up to the rostral end of the midbrain. So it includes the reticular formation and the mesencephalon and the rostral pons. And what we see with that area of the reticular formation is this is the area that is really critically important to maintaining consciousness and keeping us alert to the environment around us or within us, okay? So if we have lesions of this rostral area of the reticular formation, a very serious consequence of that is the person could lose consciousness and end up in a coma. So not being able to be aroused or to be capable of being attentive to what's going on around you. So that is a consequence of lesioning these upper parts of the reticular formation. The caudal areas of the reticular formation, again, there are exceptions to both of these, but this is just a generalization of these areas in terms of function. The caudal reticular formation has a little more variety of function. So that includes the caudal pons and the medulla. They work together with many of the cranial nerve nuclei, as well as sending projections down to the spinal cord to control, again, important motor functions like posture or tone of postural muscles. Also some reflexive activity, visceral reflexive activity that we've talked about, also motor reflexive activity. And then we mentioned before modulation of pain, and there are some centers down here that control, particularly in the medulla, that control cardiac and respiratory functions. So one thing about the role of the rostral area, the reticular formation, in maintaining alertness and the state of consciousness is that it interacts with nuclei in the diencephalon, so in the thalamus. And we see projections from these rostral areas of the reticular formation to nuclei in the thalamus that then have very diffuse projections all over the cerebral cortex. So again, the function being that by sending those projections, what you're doing is you're kind of basically waking up the cortex and allowing it to be able to now process sensory information and pay attention, be alert to sensory information coming into the cortex and make you alert and aware and awake. So that's a major function of really most of the fibers that are running up from the rostral reticular formation to the cerebral cortex. Now, some of these fibers from the reticular formation that project up to the cerebral cortex use specific neurotransmitter pathways. So they're kind of subdivisions of projections that will send signal up to the cerebral cortex. So they can have some slightly different functions based on which neurotransmitter is being used. So as we mentioned, all of them are important for alertness and attention and awareness. But for example, some of those fibers will relay information using dopamine. So those dopaminergic projections also play a role in addiction behaviors, reward, and motivation. Norepinephrine, as well as serotonin, which is 5-HT, that's another neurotransmitter, those also can be involved not only with your state of attentiveness, but they can also modulate mood and pain or arousal. And acetylcholine and histamine also play a role in increasing alertness. One thing with acetylcholine is it also has projections to memory systems within the limbic system. So it plays a role in memory as well and learning. Again, lesions to these rostral areas of the reticular formation, which is known as the pontomesencephalic area of the reticular formation, that can lead to a coma in a patient, and where the patient ends up unresponsive and cannot be aroused even to painful stimuli, okay? Some important points about the reticular formation. There are some inputs to the reticular formation that are also important in terms of the activity of the reticular formation. So for example, we mentioned that pain information coming into the reticular formation can signal descending projections back down to the dorsal horn to help modulate and control that level of pain. The other thing about pain information coming to the reticular formation is that it plays a role in making you alert, right? So you don't normally just sit there and stick your hand on a hot stove and not at least look over to where your hand is, or just not pay attention to it. All of a sudden your level of attention, your level of awareness is increased. We see that with other sensory functions as well. So for example, if you're riding up a roller coaster, right, and you get to the top of the roller coaster and you start to fall and you have that sensation, you're going to be more alert and more aware during that period, okay? And that's what's mentioned here secondly, is that you can have associations to the limbic and cognitive and emotion areas of the brain that help increase your alertness as well. So this is showing some of those outputs, those diffuse projections that basically cover all areas of the cerebral cortex by relay through the thalami, but then there's also going to be feedback from these areas as well to the pontomesencephalic area and from sensory inputs from spinal cord. Okay, so we have a little bit of time, so let's go through parts of the medulla. So what we're going to do, here comes the cross-section death march. Okay, what we're going to do is we're going to go through cross-sections of different areas of the brainstem. We'll do caudal, rostral medulla, caudal mid-pons, and then caudal and rostral midbrain. And the way that I have set this up is we'll go through tracks first, where we would find the ascending and descending tracks. Then we'll look at cranial nerve nuclei, and which cranial nerve nuclei are located within these different regions of the brainstem, and then other structures that might give us information about that particular area of the brainstem being involved in a lesion. And we'll put it all together in the figures, but we'll take them one at a time. So the way I've set this up, instead of doing sensory and motor tracks for ascending and descending, I'm just listing them as ascending tracks or descending. And the reason for that is there's an exception. Normally ascending tracks are sensory tracks, but there is an exception in the brainstem where we do see a descending track is a sensory tract, okay? So I'm just gonna list them as ascending and descending, but remember that for the most part, ascending tracts are sensory and descending are motor. So two major ascending tracts that we see in the caudal medulla that are ascending from the spinal cord are gonna be tracts carrying information about discriminative touch and conscious proprioception and discriminative pain and temperature. So that includes, so coming from the spinal cord, we have the fasciculus gracilis and cuneatus. Those are carrying discriminative touch and conscious proprioception from legs and upper limbs. And in the same section in the caudal medulla, remember what's going to happen is at the caudal medulla, there are two nuclei associated with those tracks where that information is going to be handed off. Those fibers are going to cross, and then they're going to ascend as a new track called, or a new track within the pathway called the medial lemniscus, still carrying the same information, but now it's been coalesced into one track called the medial lemniscus. So you will be able to see all of these in one section. You'll be able to see the fasciculus gracilis, the fasciculus cuneatus, and the medial lemniscus all in the same picture, in the same cross section. The distinction is these two, fasciculus gracilis and cuneatus, are carrying ipsilateral information about touch. The medial lemniscus is now carrying contralateral because that contains fibers that have crossed over, okay? And again, there is some somatotopy. The gracilis is more medial, and that's lower limb, and the cuneatus is more lateral, and that's upper limb. When we get to the medial lemniscus, it's as if the little guy who had been laying on his side is now standing straight up and down. So the lower limbs are going to be more ventrally positioned in that track, and the upper limbs are going to be more dorsally positioned. We also will see the anterolateral system. Those fibers already crossed in the spinal cord. So these are still carrying contralateral pain and temperature information from the body. And then there are two other tracks that we mentioned when we did the cerebellum, the dorsal spinocerebellar and the cuneo-spinocerebellar tract, or cuneo-cerebellar tract. Remember, these are carrying that unconscious proprioceptive information from muscle spindles and Golgi tendon organs from the limbs. So the dorsal spinocerebellar is from lower limbs, cuneo-cerebellar is from upper limbs, and they carry that information by way of the inferior peduncle into the cerebellum, which sits dorsal to the medulla, okay? So what we have here is a cross section of the medulla. So the first thing that we notice is we look at is overall shape, because that helps to tell us where we are in the central nervous system. And then we'll hone in on where the specific tracks are. So the overall shape is round like the spinal cord, because this is really where we're transitioning from spinal cord into brainstem. In terms of those ascending tracks, remember coming up from the spinal cord, we had more medially the fasciculus gracilis and the fasciculus cuneatus. So this area just peripheral to this green glob here, those are the actual tracks. Those are the axons. Those axons will relay that information to cell bodies in each of these two nuclei and hand that information off. But this is where the tracks are, okay? Just peripheral to those two nuclei. So that's our fasciculus gracilis and cuneatus. Once those cells hand that information off to the second order neurons, whose cell bodies are here in the nuclei, remember their axons cross and they will start to ascend in this track. So these are those internal arcuate fibers, those fibers that are crossing the central nervous system. And then those axons continue to ascend in this tract right here, right at the midline, right at the basal surface of the midline, right above the pyramids. This is our medial lemniscus. So again, lower limbs would be located more ventrally, upper limbs would be more dorsally in this tract. So this tract, if I lesion this tract, the medial lemniscus, I'm going to have a contralateral loss of discriminative touch from the body if the whole tract is lesion upper and lower, okay? So I have contralateral loss of touch. If I lesion the fasciculus gracilis and the fasciculus cuneatus on this side, I would have an ipsilateral loss of touch from the body because these fibers have not crossed over as they're coming up from the spinal cord, okay? And then the other major sensory tract is the discriminative pain and temperature tract. That is kind of where you would expect it to be. So remember, it was ventrolaterally positioned in the spinal cord. So as it transitions up into the medulla, we still see it in a little bit of a ventrolateral position here. This is our anterolateral tract or spinothalamic tract right here, okay? And I've tried to highlight these so that you can find them easily. Now, there are two tracks that sit out here on the periphery that are a little more dorsolaterally. Unfortunately, they don't have a circle drawn around them because they're not as distinct as some of the other tracks. So we had the cuneo-cerebellar fibers here, and just below them would be our dorsal, they listed as posterior, but our dorsal spinocerebellar tract. So remember, what's being carried in the dorsal spinocerebellar tract is unconscious proprioception from the lower limbs, What's being carried in the cuneo-cerebellar tract is unconscious proprioception from the upper limbs. Okay, so those would be out here dorsolaterally. Okay, so these are all ascending tracts. These are all carrying sensory information from the spinal cord up to the caudal medulla. Okay. Okay, now we're going to have some descending tracts. And again, mostly descending tracts are going to be motor tracts, but we're going to see one exception here as we move into the medulla. We have what's called a rubrospinal tract. Does anybody remember what that tract is?
Doesn't it control like upper extremity flexion or something?
Right. So remember that mainly innervates flexors of the upper limb. It starts at the red nucleus. It's an upper motor neuron tract. It starts at the red nucleus and descends onto lower motor neurons of the upper limb. So that rubrospinal tract, we should see that tract because it's a descending tract and it starts up at the red nucleus. That's up in the midbrain. So all these sections that we're going to look at below the midbrain, we should see a rubrospinal tract. So that includes the caudal medulla. Okay. So we have the rubrospinal tract. And then another tract that we're going to see is actually a sensory tract. That's why I have it in italics here. This is that descending spinal trigeminal tract of cranial nerve 5. Those are those fibers, they come in the pons and they descend from the mid pons all the way down to the caudal medulla, and then they will decussate and form another, or first they'll synapse in the spinal trigeminal nucleus, and then their axons decussate and will ascend back up the brainstem. Okay, so these are carrying pain and temperature from the face, but these are ipsilateral fibers. They haven't crossed yet when they're in this tract. So if this tract gets lesion, it's an ipsilateral loss of pain and temperature in the face. Okay, and their cell bodies are out in the trigeminal ganglia. Okay, another motor tract that we saw as well when we did descending motor systems is the tectospinal tract. It originates in the superior colliculus and it makes its way all the way down to cervical and upper thoracic areas of the cord. Remember, this is an important tract that takes visual information being relayed to the superior colliculus, hands it off to a motor tract going down to the upper thoracic and neck area so that you reflexively can look towards a visual stimulus, okay? And then there's a tract here that we refer to as the descending medial longitudinal fasciculus. It runs right down the medial aspect of the brainstem. Another name for this, though, is a tract that we learned already when we did descending motor systems. It's the medial vestibulospinal tract. So this is a tract that is going to arise from vestibular nuclei at that pontomedullary junction, and it will descend through the medulla all the way down into the spinal cord, again, to control truncal musculature, okay? And then the really big descending tract that runs along the base of the medulla is the cortical spinal tract, which forms those pyramids, okay? So all of these are motor tracts in terms of descending tracts, with the exception of this spinal trigeminal tract. And that's just one of the only exceptions where we see, or one of the few exceptions where we see a descending tract actually be a sensory tract. Okay, so we're back at our caudal medulla again. This is the exact same section we had before, but now we're going to focus in on those motor tracts. So the big one we mentioned were the pyramids. So this is the corticospinal tract that, again, originated up in the cerebral cortex. It's been running all along the ventral surface of the brainstem, and it is in the pyramids when it's running down through the medullary area. So that's really at the midline on either side of the midline. We also had so if this is our medial lemniscus, that was an ascending tract. If we look just above the medial lemniscus, we have the first of our, or one of our descending motor tracts, that's going to be our tectospinal tract. And then just above that, and again, these are right along the midline, is going to be that medial longitudinal, descending medial longitudinal fasciculus, which is also known as the mediovestibulospinal tract. Okay, so tectospinal, descending medial longitudinal fasciculus, okay? And then there was one exception that we had, and that was our spinal trigeminal tract. That actually sits peripheral to a nucleus that runs with it, which is the spinal trigeminal nucleus. So remember, this is carrying ipsilateral pain and temperature from the face. So if I lesion that, I get ipsilateral pain and temperature loss from the face. Remember with the pyramids, these fibers don't cross until you get to the most caudal area of the medulla. So these are carrying contralateral, these would give us me contralateral weakness if I lesion these pyramidal fibers, okay? Oh, one other one that I forgot to point out, you can always find this relative to the anterolateral tract that we pointed out before. That's the rubrospinal tract. So it's kind of squeezed between our trigeminal tract and the anterolateral tract. That's where we find the rubrospinal tract, okay? Okay, and then finally, there are a bunch of cranial nerve nuclei associated with the medulla, and many of them run the whole length of the medulla, so we will see them in the caudal medulla as well. So one is the hypoglossal nucleus. Remember, this innervates muscles of the tongue. We have the dorsal motor nucleus of the vagus, which runs with cranial nerve 10. What kind of fibers are carried in the dorsal motor nucleus of the vagus? So we could think about what kinds of deficits would we have if we lesion that nucleus. Those are our parasympathetic fibers, right? So if we lesion that nucleus, then you're going to have visceral effects, right? Because that's the vagus, those are the fibers that run with the vagus nerve down to the thorax and the abdomen, okay? The nucleus ambiguus, that runs the length of the medulla. Remember, those are those fibers that are going to be our special visceral efferent fibers. They run with cranial nerves 9 and 10 to innervate skeletal muscle, particularly of the larynx and pharynx. And then we have the solitary nucleus. This is that nucleus that receives taste fibers, as well as those general visceral afferents about blood pressure from the aortic arch and the carotid sinus. So the rostral part of that nucleus will carry the taste fibers from 7, 9, and 10, and the caudal part will receive general visceral afferents from 9 and 10. And then the spinal trigeminal nucleus, this is a nucleus that's going to accept sensory information about pain and temperature. Most of that information is coming in by way of cranial nerve 5. But remember, cranial nerve 7, 9, and 10 can also send pain information about that little strip outside the ear and hand it off to this nucleus as well. So if we look at where these nuclei are going to be located in the brainstem, again, here's our same cross section that we looked at before. Cranial nerve 5 we can easily find because remember we already looked at the spinal trigeminal tract, which sits peripherally. So more just medial to that tract is going to be the spinal trigeminal nucleus, okay? And again, that's ipsilateral pain and temperature. Now the others are easy to find. Again, if we look right at the midline, here's our central canal. We've already identified these structures, our medial lemniscus, our tectospinal, our descending medial longitudinal fasciculus. And then right above that, right at the midline on either side is a large nucleus that is the hypoglossal nucleus. And then we just start marching our way a little bit dorsolaterally. So we have first hypoglossal nucleus, then we have our dorsal motor nucleus of the vagus, and then we have our solitary nucleus. Okay, so those three are kind of right near each other, and we just kind of march our way out dorsolaterally. There's one more nucleus that runs the length of the medulla. That was our nucleus ambiguus, and that sits really kind of right almost in the middle of between the dorsal and ventral surfaces of the medulla, as well as right in the middle between the medial and lateral aspects of the medulla. So it's kind of right here in the middle of the tegmentum that we see that nucleus ambiguus, okay? One thing that helps you figure out that this is, or it may help you figure out that this is the hypoglossal nucleus, is you see these fibers that are leaving that nucleus, and they are exiting the brainstem down here on the ventral surface of the brainstem, just lateral to the pyramid. And that's what we see with the hypoglossal nerve. It comes out lateral to the pyramid, okay? Okay, some other things that don't fall under those categories of ascending, descending, and cranial nerves are some structures though that are unique or that are present in the caudal medulla, but also may be unique. Not all of these are unique to the caudal medulla, but some. One thing that we see is the ventricular system is very narrow. So that does tell me I'm in medulla, okay? Because I have this little central nucleus or ventricular system. Now, that's also true of the rostral medulla as well as the caudal medulla. So that may not allow you to distinguish rostral from caudal medulla, but it might help tell you that you're in medulla. Reticular formation, we're gonna see that throughout the brainstem, so that's not a very good localizer for us. But one thing that is really a good localizer are these two up here, the nucleus gracilis and the nucleus cuneatus. Those are only present in the caudal medulla. So not only does this tell us we're in medulla, but it tells us we're in caudal medulla, not rostral medulla. Okay, so this is a really important localizer for caudal medulla. We can see parts of what's called the inferior olivary nucleus, but quite honestly, we will see this much more clearly and better when we get up to rostral medulla. So this is actually a better localizer because it's more obvious in the rostral medulla, okay? So if we look back at our schematic again, these are our nucleus gracilis and nucleus cuneatus. And again, we only see these in the caudal medulla, and they'll be here on the dorsal surface, just deep to the fasciculi, okay? Sometimes you can see little bits of the olivary nucleus in the caudal medulla, but again, I'll show you on a silver stain section. It's usually not quite as obvious as what we see in the rostral medulla. And then, as we mentioned, reticular formation really just exists in this area and here, all through here, it's diffuse. So that's not a great localizer, and it's present in all parts of the brainstem. And then the other thing is that really small ventricular system here, together with some of these other features that we've mentioned, okay? So those are important anatomical structures and pathways. This is just showing a silver stain section of the same area. So remember, with a silver stain section, what we have is what's showing up dark are myelinated axons. So fiber tracts are going to be dark on a silver stain section, but areas where we have cell bodies, those are going to be lighter. Okay, so those will be nuclei. So we can look at some of the tracts and the nuclei. Again, on an exam, I usually will not go in and label something on a silver stain section unless it's really very obvious, like, for example, the nucleus gracilis or the nucleus cuneatus, something like that. But I wouldn't, for example, tell you to point out reticular formation or the rubrospinal tract, because it's just very difficult to see, okay? Mostly what I would use these for is just having you identify the area of the brainstem. Is this medulla? If it is medulla, is it rostral medulla or is it caudal medulla, okay? That's usually, if I use these, that's what they would be for. But we can see some of the things that we talked about. So that small central canal of the reticular system. Again, we can see very clearly the pyramids here on the ventral surface right at the midline. Just above those pyramids, we see these dark fiber tracts that make up our medial lemniscus. Again, hard to decipher, but we know just in relation to the medial lemniscus, we would have our tectospinal and our descending medial longitudinal fasciculus. And remember, just above that area, you see this light patch here that is going to be our hypoglossal nucleus. Okay. Now the other nuclei again are going to be a little bit harder. They're all light, like the hypoglossal nucleus. Again, they usually kind of march your way dorsally. So things like the dorsal motor nucleus of the vagus, the solitary nucleus. Again, that's not something I'd ask you to pick out on the silver stain section. It's just not very clear and very easy, but you should know on the schematics that we just did where those should be relative to one another. And again, nucleus ambiguous would be kind of right here in the mid area. And then we had our spinal trigeminal nucleus here more medially, our spinal, well, you can see that better over here. Spinal trigeminal nucleus, which is lighter. And then we see these dark staining fibers as our spinal trigeminal tract. We have our anterolateral system kind of down here, ventrolaterally in this area. Right above it is where we would find our rubrospinal tract. Yeah, I think we've covered most everything here. Again, this is showing how that inferior olivary nucleus is really not very clear and demarcated in the caudal end. Okay. Okay, where are we? Okay, I'm gonna stop there today because it's been a lot of information. when we get together next time, we'll finish going through these sections. So we're going to keep moving up to rostral, medulla, pons, midbrain, and again point out these structures. Now, when you are getting closer to taking the exam, I will post a study guide, and on that study guide, it will list the specific structures that you are responsible for knowing, but most of them are the ones that I have here in the PowerPoint slide. Okay, so as long as you know those structures that we've gone through on the PowerPoint slide, you should be okay. Okay, and again, that's only part of the exam, is going to be having you actually be able to identify, first of all, what area of the brainstem you are, identifying some specific structures, particularly structures that help localize you to that particular area of the brainstem. But half the exam is going to be, okay, now that I know these structures, I know what the function of these structures are, what happens when I lesion this area of the brainstem? So what are all the structures in that area? Do I have a collection of signs and symptoms that fit the anatomy for that area, the caudal pons, or is this the deficits that I'm seeing fit better with the rostral midbrain? Okay, and again, you want to look at as many things as you can. So you don't want to make your diagnosis necessarily just on a particular cranial nerve, but you may want to look at things like the tracts and other structures that might be present in those areas. Okay, so that's part of learning how to localize to brainstem. Okay, okay, any questions? Okay, well, I will see you guys